Thank you. Okay, so good morning, everybody. My name, as Alejandro said, is Laura Camelo, and I am doing my PhD right now in McGill University in Montreal. And I'm going to share with you my project, which is focused on linking gut viral populations to bacterial colonization and malnutrition. So the human microbiome is home to many different microorganisms, among them bacteria, phages, fungi, and protozoa. The gut microbiome helps the human host with different functions, for example, nutrient uptake, immune system modulation, and to prevent pathogens from colonizing. This gut microbiome establishes very early after birth, and it follows a really well-defined pattern in which bacteria and phage communities both mature as the infant ages. We would like to focus on phages because they are drivers of bacterial diversity at different levels. They can kill bacteria and they can also modify their genetic pools. So we expect them to be to have a huge impact on gut bacterial colonization and succession patterns. Thus, we can use the microbial composition, both phages and bacteria, to predict the age of the infants. So there are two studies here that I show in which they predicted the age of the infants based on, can you see my mouse? Not really, right? There, okay. So here in the left is this paper where they predicted the the age of the infant based on the microbial composition, only bacteria, and then they correlated it with the actual age of the infant. And as you can see, there is a nice correlation between the two of them, showing that there is maturation in the gut bacteria community's composition over time. Then in this paper, which is from Alejandro Reyes, they also, instead of predicting the age based on bacterial communities, they did it with phage communities, and they also showed that there is a progression of phage composition as the infant ages. So then we wanted to see how does a condition like malnutrition, in which nutrient uptake is being disrupted, how this affects this normal microbial maturation. We're also very interested in malnutrition because it affects 1 in 4 children worldwide, and it has not only long-term consequences in the individual's life, but also their future generations. So there is this paper, also the one with the bacterial composition maturation, in which they show that for children with severe acute malnutrition, when we predict the age of them with their microbiome, the age is less than their actual age. This is suggesting that the microbiome is immature in children with malnutrition. And it also stays like that even after dietary treatments. So then what we would like to focus on is to see what's the role of phage bacteria interactions in the gut maturation. Also, there have been phages that have been associated in malnutrition. However, this has not been really well studied, so we don't know what is the role of these phages in the maturation of gut viral communities. Also, phages evolve rapidly, and they harbor a lot of genomic variation that also supports bacterial community structure. There is this paper in which they show that there is a lot of genetic heterogeneity in grass phages populations between infants and their mothers, suggesting that genomic variation might be a factor that is important for gut maturation from infancy to adulthood. And then also genomic variation or microdiversity affects phage fitness. For example, increasing the host range or causing host jumps that might be important for this gut bacterial maturation as well. So further research is needed to understand how this microdiversity shapes phage host dynamics during early life development. So we hypothesize that phage host interactions and genomic variation are key to the maturation of bacterial communities in the gut, with effects observable at the subspecies level, and these patterns are influenced by undernutrition. To test this hypothesis, we stored these patterns in a cohort of 232 children from Zimbabwe, sampled from birth to 650 days, and this included stunted and non-stunted controls. So here in blue, I have the healthy children that are 172, stunted children that are 29, and then we have 31 children that are transitioning from healthy and stunted states. This is the analytical workflow that we followed. So using the reads that we got from the publicly available data set that I just described, we followed quality control and reads cleaning, then the contaminated the human reads, and then we did an assembly of the bacterial and viral context using metaspades, and only to focus on the viral portion, we use GenoMap as the viral identification tool. Then we wanted to replicate this data at the subspecies level. So for that, we did an all versus all an all versus all comparison of the average nucleotide identity with all the viral context that we had, and that gave us a natural gap in the ANI at 99.5% and 95% 99.5% identity and 95% coverage, defining these as groups of phages of the same strain, which we also call viral populations or VPOPs for short. Then we did read mapping for ecological analysis, host prediction with IFOP, and microdiversity calculations with each strain. So for our first result, we got as we expected, that the infant virome is highly individual, and as it is shown in this graph here, the comparisons between infants and these distances are higher than the distances within the same infant, but over time. And then looking further into this, we saw also that the distances increases as the infant ages, meaning that, yeah, the virome is divergent and is changing as the infant is developing. So we wanted to know how are these gut viral communities maturing in all these nutritional statuses. So what we did is that, we built a random forest model to predict the age of the infants using the abundance matrix of our viral populations in healthy infants. And then we tested it in stunted and transitioning infants. So what we got is that here in blue is the healthy maturation curve, which is different from the curves that we get, for example, for stunted and transitioning data, transitioning infants, in which we can see sort of a delay in this gut viral maturation. Then we wanted to see who are these viral populations affecting, because we don't know their hosts from previous studies. So what we did is to get the host information from IFOP, and then we use that as a starting point to correlate phage and bacteria colonization patterns in early life. So using the bacterial host family, which we call phage host families now, these are groups of phages that infect the same family, bacterial family, and we did the maturation model again with the random forest, and as we can see, it mimics what happens with the viral populations in which the healthy development is kind of like more accelerated than the stunted and transitioning ones. And then using a tenfold cross-validation, we looked into the 18 most important PHFs or phage host families that correlate with this gut viral maturation. And then we plotted that among different ages in healthy children. And as you can see, we see some patterns of late colonizers, such as these ones in the yellow box. And then in the blue box, we see the earlier colonizers. This is really mimicking or mirroring what is happening in bacterial colonization models, in which Bifidobacteriaceae are like early colonizers, and Faecalibacterium and Dorea are associated with later colonizers, which also associates with changes in the oxygen levels in the gut. Then we wanted to see if there were any difference at the genomic level, if there were any genomic variation changes that were different between the nutritional statuses for each of these important phage colonizers. So we did the same random forest model to predict phage, the age of the infant, but in this case, we didn't use the abundance of the viral populations, but rather the nucleotide diversity. The nucleotide diversity is a metric of the genetic variation of these strains in each time point. And then we tested these random forest in stunted and transitioning infants. So as you can see, we can also get a maturation curve for both the three statuses, but this time we don't see any differences between them. And then also our model is also less accurate. So then we wanted to see what is happening at each PHF level. So how are these families accumulating diversity over time? And we focus on this, for example, for families in which their phages are accumulating diversity differently in the three different nutritional statuses. However, these changes are not significantly different, but we can see some interesting trends. So to conclude, I would like to say that there is, well, our work demonstrated that there is a maturation of gut barrier communities during early life, that we have early to later colonizers, same as what we have with bacterial colonization, that this gut viral maturation is impacted by undernutrition, and there is potentially a delay in this viral colonization. We need to further study this to see if there are viral populations that are discriminant or static. So, for example, if they are only present in stunted children, and also how does this phage colonization affects bacterial colonization patterns. And then regarding the nucleotide diversity, we also showed that it matures with age, as well as the abundance patterns. And this maturation is more correlated with age than it is with the nutritional statuses. And but we do see some trends that are different between some of the nutritional status for some of the families, phage host families. Then we want to see where are these genomic variation happening, if it is in genes that are related to phage host interactions or the phage replication itself. And then same as what we want to have with the viral populations, we would like to see if there are some phages that are infecting families that only are related with stunting, and how are these varying over time. So with that, I would like to thank my lab, especially my PI, or Professor Cory Maurice, and my colleague Michael Shamash, who has helped me with several sample collection and also some analysis discussion. Also my community members, Dr. Jessica Piro and Dr. Alejandro Reyes. And thank you everybody for listening to me, and also for to the ISCB for making this presentation happen today. That's it.
We have time for a couple of questions.
Thank you for your talk, Laura. I was wondering, so bifidobacterial phages are known to be important for the establishment in the early stages of life. And did you find some bacteria and bifidobacterial phages in those malnutrition samples, or could you classify those to that level?
Yeah, we do see some of these bifidobacteria phages here. Sorry that it is not like super big, but yeah, we do see it in the three statuses actually. So healthy is in blue, yellow would be stunted, and then in green transitioning, and it is one of the more like of the families that have the most data. So yeah, it is very prevalent.
They decrease if the malnutrition is high?
I don't see like because we don't have any statistical differences in this data, but yeah, like you can see a little bit that the yellow boxes have like a different behavior than the healthy and transitioning boxes. However, this is not statistically significant, so we would like to see further in this data. Also, this is bulk metagenome data, and we do have now a new cohort of children from DACA from which we could extract the free particles, the free viral particles, and then look at, for example, in the active viral portion, what is happening, and if there is more variation between the nutritional statuses in that data, in those data, more than in this data that is bulk metagenome.
Any other question for Laura?
I have just a small question. You were showing the dynamics over a range of time.
The heat
But
map.
for me, wasn't totally clear because you said age and it was like 200, 400. So,
This one?
no.
This one?
Further. That's weeks.
Ah, sorry, this is days. I'm so sorry. This is the days. Sorry, yes.
I have a short question. I don't know if I should wait for the committee meeting or do it here.
As you wish.
But so you say that, and we have seen that the fish community mimics the microbial community, but that would imply in general that fish community you're seeing is mostly temporal phages, or probably the role of the medic phages in the dynamic.
That is actually one of the questions that I find more interesting. And this is because this is both metagenomes, I have mostly like prophage dynamics than what I would have with free virome data, in which I can see which viruses are being induced and how are they progressing over time. But I don't have the data. I do expect, for example, that virulent phages would accumulate more in diversity than prophages because they are replicating faster than the prophages. That vary at the same range as the same at the same rate as bacteria, but do whatever. Yeah.
Thank you, Laura.
Thank you.